Hello, I'm Lucy Godley from the University of Chicago, and I'll be speaking this morning on hereditary predisposition to myelodysplastic syndrome. My disclosures are there at the bottom of the slide. I do receive royalties from a chapter that I wrote on this topic for UpToDate. So I'd like to begin with acknowledgements, just thanking my patients and their families for showing me a topic that's underappreciated in our field, but hopefully is emerging in appreciation. Everyone in the lab who works very hard on this topic, and Soma Das in our genetic services lab, and Soma and her staff were willing and did put together the clinical testing pipeline that I'll share with you today. In our Department of Pathology, Jeremy Siegel and Jim Vardaman also incorporate germline predisposition into their assays. So I have the idea that many of you do, that if we think of patients at a very deep level, we can really develop effective treatment strategies for them. And so for me, realizing this goal of precision medicine in oncology begins with defining baseline genetics. I don't want to overemphasize this, though, in the sense that I, of course, appreciate acquired genetics are very important. And those are layered on top of the baseline genetic changes that we have. But we will focus in the next 20 minutes or so on baseline genetics. So this is the current table that I use to outline all the different genes and disorders that confer germline risk to hematopoietic malignancies. On the left is a column of genes that confer risk to myeloid malignancies only. Next column shows you genes where deleterious variants confer risk for lymphoid malignancies or immunodeficiency. And I do have a bias that any germline predisposition to immunodeficiency will eventually be shown to confer risk to lymphoid malignancies. In the third column, you see the genes that predispose to hematopoietic malignancies, both myeloid and lymphoid. And finally, in the last column, genes that confer risk both to hematopoietic as well as solid tumors. Now we have a riddle that goes, how much does a ton of feathers weigh? And of course, the answer is a ton. You've given the answer just in the question. But the paradox is that each feather is very light, but when you have so many of them in totality, you have something very heavy. And similarly, we may say that each one of these disorders is relatively rare, but there are so many of these disorders that in totality, you have something common. And so I've tried to show this in a pictorial way with these larger feathers at the bottom. For example, bone marrow failure, dyskeratosis congenita is comprised of many, many genes. And so again, we may think of each disorder as somewhat rare, but there are so many to consider that this is a very general phenomenon. These are the criteria that we use currently to decide whether a patient deserves clinical testing for germline predisposition. First, the personal history. If an individual has multiple cancers, then we recommend germline testing. And we've thought of therapy-related leukemias, those leukemias that arise after treatment for a solid tumor or after cytotoxic therapy for an autoimmune condition. But actually, we have more patients who survived a hematopoietic malignancy and then go on to develop their solid tumors. We call those double cancers. And whether the patient gets the leukemia first or second relative to the solid tumor makes absolutely no difference. The testing positivity rate in patients with multiple cancers is about 20%. Secondly, if we see a heme malignancy diagnosed at a much younger age than what's expected from the general population. And here, relative to our topic today, we'll talk about young MDS, and I'll show you our data in that regard. Now, I think people accept very easily that if you present at a very young age relative to the average, you have a risk for germline predisposition. But I think people have taken this idea to then mean that if you present at an average age for a particular diagnosis, the germline is not contributing. And I think that's absolutely wrong. And germline DDX41 shows us that. And so I think we've missed a lot of germline predisposition because people still present with their diseases in an average age. We also pay attention to the family history, and we define a positive family history to be an individual with a heme malignancy who has another individual in the family with a heme malignancy or a solid tumor diagnosed at 50 years old or younger, or if there are other heme abnormalities within the family, either macrocytosis, bleeding, anemia, et cetera. Obviously, those are more difficult for people to know about relative to tumors, but we always ask. Now, this is the algorithm that we use for the patient workup, starting with that personal and family history, which we take every time we do a bone marrow biopsy on a patient. And we do that because at the time of the bone marrow biopsy, it's very easy to get the skin biopsy, which we use preferentially for this testing. We pay a lot of attention to the molecular profiling being done on the leukemias because many of the genes that are mutated as somatic events can be mutated as germline events, and you really can't tell from the results of that molecular profiling whether that variant is somatic or germline. We'll talk about that. And then also we pay a lot of attention at the time of related allogeneic stem cell transplantation. So we perform that skin biopsy, we grow the skin fibroblasts, isolate genomic DNA, and then right now, our clinical pipeline is an augmented whole exome sequencing platform. It's augmented because we add in probes that pull down non-coding regions, promoters and enhancer regions where we know deleterious variants can exist. And we use very special bioinformatic analysis to detect single nucleotide variants and copy number variants. And I want to emphasize how common germline copy number variants are. And most panel-based testing will miss copy number variants. So you have to be very careful to make sure that your platform is comprehensive. If we get a positive result, we do cascade testing throughout the family if they desire. And if negative, then the individual and family goes into our research collection, and we're defining new syndromes all the time. What's our yield? Well, you can see we test throughout the entire age range of life, and the diagnostic rate is about 20%. This means that the criteria I shared with you are not even broad enough. This is too high of a percentage from a human genetics standpoint. We want a positive testing rate more of about 5%. Now, I said that we use our somatic or molecular profiling panel to find families like this. We don't rely on that testing. As I shared with you, we have a very particular germline testing platform. But if we see a variant on somatic testing, we do consider whether it could actually be germline. Now, I'm going to first explain why we don't rely on those somatic or molecular profiling tests. Then I'll go on to show you how we actually do use them. So first of all, we've already talked about how we're still discovering more and more germline genes. That means our platform for germline testing has to be able to expand over time. On the somatic side, I would argue that we have largely defined the acquired mutations that accompany heme malignancies. That means our platform can be stable and we can just look on those exons for those somatic variants. Now, from a biological standpoint, the reason that we don't want to rely on these molecular profiling panels is that these are being done on hematopoietic tissue, and hematopoietic tissues undergo somatic reversion relatively easily. So if you get a negative result, that still could mean there is a germline mutation. If you see something, it's definitely there, but if you don't see anything, there still could be something there. This is why SAMD9 and Z9L were missed for decades, because people kept sequencing these individuals' blood and bone marrow. So again, mutations are prognosticated based on exons that can be paneled very easily. In contrast, for the germline, we need to assess copy number variants and non-coding regions. So an exome isn't good enough. You have to either do an augmented whole exome, as we discussed, or a panel with a microarray to be able to capture those copy number variants. And now just thinking about particular alleles, there are certain genes and certain alleles that we've only seen as germline alleles. So those are particular alleles in CHEK2 and DDX41. We can just look at the allele and we pretty much know that it's germline. For most genes, though, you cannot look at the allele itself and know whether it's somatic or germline. The same exact allele can be somatic or germline, so you have to do additional testing. And I would say even for CHEK2 and DDX41, we always do the skin biopsy to document that it's a germline variant, although we would be very clear with patients about how confident we are that the variant is likely to be germline. So now that we've given those caveats, let's look at how we use clinical testing from leukemia cells to find these families. And of course, the idea here is that we're doing this molecular profiling systematically over time to be able to define molecular remission. And somatic variants disappear as people go into remission, but of course the germline variants are always present. So first, I want to show you an example of where we use this kind of analysis to find such a family. So here we have my patient with acute myeloid leukemia diagnosed at 78. His mother had breast cancer at 50 and two uncles with lung cancer who were smokers. And I'm kind of giving you the answer here that he's going to end up having Li-Fraumeni syndrome, and you absolutely would not expect that from this family history. And I think that's very important, especially for Li-Fraumeni. All the individuals we've identified with heme malignancy with Li-Fraumeni syndrome have pedigrees like this that do not fulfill any clinical criteria for that syndrome. And so how did we figure it out? Well, when we did that molecular profiling at diagnosis, he had a DNMT3A variant at 36% VAF, and the TP53 was present at 64%. But at remission, that DNMT3A went down to clonal hematopoiesis levels, whereas the TP53 remained high. Then we did the clinical testing, and yes, documented this is Li-Fraumeni syndrome as we suspected. So this sequential analysis over time is very helpful to identify these individuals. What percentage of deleterious variants in genes that can confer germline susceptibility and show up on these somatic panels are actually germline? Well, when we did this as a study, all of the germline variants had a VAF over 40%, and about 25% of these pathogenic variants were actually germline. Now, since we've done this study, this VAF has crept downward. So recently we had a patient with a germline BRCA2 variant that on this panel came back with a VAF of 31%. So these, there is technical variability, of course, in all assays. And so now we prioritize any variant allele frequency over 25% to be potentially germline. We've discussed this already, that most genes you cannot look at the allele and say or predict whether it's germline or somatic, whereas for CHEK2 and DDX41, you really can. For example, in our hands, all truncating mutations in DDX41 are germline. Several mutations are particularly common in certain populations, and there's one mutation that's a hotspot mutation. Let me just go back and state what those are. The common mutations in particular populations would be the A500 frame shift in Asian populations, especially Koreans and Japanese, the D140 frame shift, and the M1 question mark in Northern European populations. And the common hotspot mutation is the R525H. So if you do molecular profiling and DDX41 is on your molecular profiling panel, and you see an R525H in the context of a truncating mutation, really think about the germline. So we plot these variants allele frequencies over time, and we actually calculate a variance of the VAF with a very small variance like here being germline and large variances being somatic as people come in and out of remission. These two individuals, like that other person I showed you earlier, have Li-Fraumeni syndrome, and their family histories would not fulfill, or personal and family histories would not fulfill any criteria that have been described. So I think we have to be very careful with Li-Fraumeni syndrome when we come from a heme malignancy perspective. In contrast, BRCA2 is most often a germline variant. Sometimes it can be somatic, but mostly it's germline. Now, how often do people actually have germline predisposition? We asked this first for myelodysplastic syndrome, since of course this is an older person's disease. We wondered, why are these younger people getting MDS? So because I'm an adult oncologist, we arbitrarily chose the ages of 18 to 40. I've already stated that our gold standard is cultured skin fibroblasts, although many of these individuals had only bone marrow aspirates for the sample available for research. And so for those, we cultured the bone marrow-derived mesenchymal stromal cells, which are non-hematopoietic and serve as a germline sample. In a very small number of cases, we use saliva or paraffin-embedded tissue, both of which are obviously contaminated with hematopoietic cells, and T cells are obviously hematopoietic. This was a very large study with samples collected from all over the world. So I'm very thankful to all of these collaborators to allow us to put together a cohort of about 120 individuals, both myelodysplastic syndrome as well as aplastic anemia, given the diagnostic overlap between those two entities. And the variant calling here was very conservative, and likely pathogenic or known pathogenic variants were found at about 20% in the MDS patients and 15% in the aplastic anemia patients. Now, unbeknownst to us, the pediatricians were doing the same experiment. And if we put the data together across the age spectrum, what we see is that if you present with MDS as a very young child, say less than 5 years old, you're most likely to have a mutation in SAMD9 or D9L. If you're a teenager into young adulthood, most likely got a 2, especially if there's deletion 7 in the MDS. And adults, young adults from 18 to 40, we see deleterious variants in DNA repair and telomere biology genes. And we've already discussed how DDX41 presents in the elderly. You can see that we have a gap here in our knowledge from 40 out into the older age range. And that's a gap that we're actively trying to fill right now. So I like to say age of presentation is a surrogate for the pathway. The age at which you present merely is a clock of how quickly that germline predisposition gives you MDS. So I just want to say again, we've only had time to talk about baseline genetics, and obviously overlaid on top of baseline genetics are the baseline epigenetics that someone has and the acquired genetics that someone accumulates in their hematopoietic stem cells, in their other organ stem cells and tumor initiating cells in their body, many of which bring the acquired epigenetic changes. And I think once we put all of this information together, we'll really be able to develop effective treatment strategies for people. So with that, I'm thankful for your attention and happy to take your questions.